Hey, welcome back, guys. It's set 11 amino acids and proteins. This is as biochemistry like as we can get. So, we're going to talk about amino acids and how they combine to make proteins, the different formations, if you will, and ways that proteins put themselves together. And then we're going to talk a little bit about protein reactions, hydrolysis, and denaturizing or denaturation, denaturalized, denatured. Okay, so let's get started. Proteins are major building blocks for all things alive, biological activity, biological species. Proteins all have to do with different cellular functions, and cells are the essence of life. So we've got structural proteins, contractile proteins making muscles move, actin fibers, myosin fibers. Collagen and keratin are two things that I know you've heard about with just shampoo commercials and skincare commercials. Transport proteins, everybody knows hemoglobin is in the blood, and then lipoproteins are something that we're going to study. Storage stores nutrients. Casein is the protein that's found in milk. Ferritin contains iron and is stored in the spleen and liver. Hormonal proteins like insulin and growth hormone, human growth hormone. Enzymes, sucrase we already talked about. Trypsin, we talked about other ones as well. And then protection proteins, immunoglobulins, to stimulate immune responses. Boy, this sounds like something very important during the COVID-19 crisis, right? Okay, so these are different classifications, 1, 2, 3, 4, 5, 6, 7. Now let's talk about how proteins are made. Proteins are natural polymers, meaning polymers that are found in nature. Poly means many. Mers is sort of units. Okay, so amino acid units join together by what's known as a peptide bond. There's the amide bond between that's an amide group, and this is considered to be the peptide bond or the amide bond. And the specific order of those amino acids in the chain determines how the protein reacts with other proteins in terms of whether or not they fold over, stay straight, and part of that has to do with how the protein then will function in the body. Amino acids are the building blocks of proteins. They're the monomers that get linked together to make the protein chain. There are two functional groups on every amino acid. On that alpha carbon, that's the alpha carbon. You've got an amino group, and you've got a carboxyl group. And these are the same. In fact, this part is the same on every amino acid. That's sort of how you define it as an amino acid. It's not a functional group. It's bigger than that. It's essentially a biological molecule. And the side chain changes and therefore makes a different amino acids. There are 20 common amino acids that are found in nature. And no, you're not going to have to memorize what they are, and you're not going to have to memorize what their formulas are or their names. These will be given. But your job will be able to pick them out. You must, 1, pick them out, 2, categorize them. and we'll go through the different categories. So I'm not going to ask you to memorize the formulas, the compounds, or their names, but you'll have to be able to look at a long or some amino acid linked up protein and be able to identify where the different amino acids are, be able to perhaps do reactions with them, and then be able to categorize them as well. At physiological pH, meaning pH of the body, amino acids are ionized. Now, what do we mean by ionized? I think it's obvious what we mean by ionized. It means they form charged species. Actual charges, +1 or -1. Okay, so how does it work? Well, a proton gets transferred from the carboxylic acid part to the nitrogen part. To give the carboxylate ion, that's carboxylate, and an ammonium group, which is that part. Okay, so at physiological pH, no matter what the R group is, this is what you have, which is both species with ions at the same time. It is a compound that has both a plus and a minus, and that's called a zwitterion, which is really sort of one of my favorite words as far as chemistry words go. I mean, zwitter. How many times do you get to say zwit all at the same time? So, losing my mind a little bit as we're all the way to set whatever this is, 137. But zwitterions are a neutral compound, so the compound total overall is 0. Okay, but within the compound itself, there is an absolute +1 charge and a -1 charge. The neutral form does not exist at physiological pH. Instead, we consider this a salt because it's got plus and minus in it. It's got ions, and it's the neutral form of amino acids. Okay. The simplest amino acid, the simplest one, is called glycine, where R is just an H. The R group is called the side chain, so this means amino acid, and all of them have that. And the only thing that we look at to change is this, the R group. When R is an H, then the amino acid is a glycine, and that's a molecule all by itself. And that's the only thing, like I said, that varies between the different amino acids. And that R group determines what it is. It determines the identity and the type. Okay, 19 out of the 20 amino acids are considered primary amines with only one R group. There's only one secondary amine with 2 groups, and that's proline, and we'll look at these in a bit. All amino acids can be categorized as either nonpolar with hydrocarbon side chains, polar, neutral with polar side chains, polar acidic with carboxylic acid side chains, and polar basic with amine side chains. Okay, those are the 4 different possibles. Now let's look at them in particular. For the nonpolar amino acids, the R group is either H, an alkyl group, which is nonpolar, or aromatic, which is also nonpolar. So you can look at the R group itself and decide, oh, the R group is nonpolar, and so therefore there's no charge involved at all. And so we always have a neutral charge because of the Zwitterion part. Okay? So these are the nonpolar amino acids. And they're all hydrophobic because of the nonpolar R group. All right? Now, is the amino acid in total considered to be hydrophobic or just the side chain? Well, what do you think? Hey, this is a salt, isn't it? And this is not a very large R group, right? And so amino acids, even these don't have that many carbons. So these are all soluble in water. Are you surprised by that? Are you surprised that amino acids would be soluble in water? No, we need them in order to be incorporated into our cells, into our body. We eat these things and then incorporate them into cells. So of course, amino acids are soluble in water. We're just talking about whether or not the side chain itself is nonpolar and the side chain is neutral. Okay, so we've got glycine with an H as the R group, alanine, which is a methyl, valine, which is an isopropyl, leucine, which is a isobutyl, isoleucine. No, I'm sorry, this is a secbutyl. This is a isobutyl. There's phenylalanine. There's methionine, the sulfur. I've got proline. There's the linked up in two spots. That's why this is a secondary. I should probably write that as a secondary amino acid. And then we've got tryptophan, which looks like that. Okay, so again, I'm not going to ask you to memorize these. I'm never going to ask you on a test. Write down the formula for tryptophan, or write the amino acid triplet of alanine, valine, methionine. No, never going to happen. Unless I give you a chart that has all of these showing and their names, and you can go find the amino acid, and then your job will be to link it up in the correct way. Okay, so relax. It's not going to be so bad. I'm going to give you information. Now, if we have a polar neutral amino acids, the R group has to be polar and it has to be neutral. So alcohols are polar and uncharged, right? As are thiols, as are amides. So serine has that as the R group. Threonine has that as the R group. Tyrosine, cysteine, asparagine, and glutamine. These are all neutral, no charges on them, and clearly they're polar, right? Polar acidic and polar basic are different in that we have a carboxyl group that is the R group and it has a charge. Notice that they have charges. So an acidic, as you can see, is going to have a carboxyl group attached. Either aspartate or glutamate are the two different amino acids that are acidic. And there's three possible basic. Histidine, there's a charge. Lysine, there's the charge. And arginine, there's the charge. Okay, so again, we're not going to ask you to memorize these, but you would need to be able to recognize whether or not it's polar or not. And of course, these are all polar. How much more polar can you be than if you have an actual charge on you? And then you have to decide if they're acidic or basic. And I think that's pretty easy. All the basics have NH3 plus involved, right? Or some RR version of it. And then carboxyl group is the other side. And you will always associate carboxyl groups with an acidic compound and amino groups with a basic compound. So these are pretty easy to categorize. And you can see those charges that are on those amino acids. And that becomes important whenever they're part of the protein itself. All right, good. So let's identify these things and decide as polar or nonpolar. So in this case, we flip this one over, right? Actually, the whole thing is sort of flipped backwards, but it doesn't matter. This is the R group. This is the R group. Since it's H, that's nonpolar. And if you go back, however many it takes to go back, you'll be able to find which amino acid this is, right? You would just look for the R group that is associated with it, and you'd look and you'd look and you'd search, and you would find, what would you find? You'd find glycine. For A. And you would look and you would look and you would look, and you would decide, hey, is this a polar group or a nonpolar group? Well, sure, it's polar. And which one is it? It's threonine. Okay, so those are the amino acids. Do some practice, see if you can do that. It's not too bad. Just match them up. Okay. All right, so now let's talk a little bit about how the amino acids link up to make proteins. And the linkage is a peptide bond. Peptide bond is an amide bond. It forms between the carboxyl group of one amino acid and the amino group of the next. So we're going to make an amide peptide linkage. How does it work? Well, the minus and the plus sort of want to react. There it is. That's the amide bond. You're going to have the free amino end is always written on the left. That's why they call it N-terminal. It's on the left. And the free carboxyl group is called C-terminal. And it's written on the right. And we always write them that way just for convention. And a peptide is two or more linked amino acids. Okay. So peptides, which are small, and then proteins, which are longer, are formed when amino acids are joined together by those amide bonds. A dipeptide has two, a tripeptide has three, and polypeptides have many, 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 many. Whereas proteins have many, 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 many amino acids linked together. Okay, so if you want to identify any compound as a peptide or as a protein, or to decide just how many amino acids are in the compound, you have to just count up the number of amides, which means you need to identify those amides. Okay? And then you know how many different amino acids are involved. N-terminal is always written on the left-hand side. C-terminal is always written on the right. Peptide structures are always written with the N-terminal to the left. Always, always, always. And this brings in a little bit of the naming that's involved. And since we, for sure, always go left to right when looking at proteins, if you're from this country, it's like reading a book. English, I don't know. I don't know the number of languages that go right to left. Couldn't tell you. I just know mine, which is English. And we all know that we go, when we read a book, we go from left to right. It's the same thing with proteins. So you're going to look at this as the first and that as the second. And that's why that's how the naming goes. And then it's opposite, going the other way, because we always start with the N-terminal and end up with the C-terminal amino acid. Okay. So how do we draw a dipeptide? We're going to draw val-gly, and then we're going to label the N-terminal and C-terminal amino acids. So step 1, draw structures or copy down structures of the individual amino acids from left to right. So since val is first, that's valine, and gly is second, that's glycine. So we write valine and we write glycine. Notice that N to O or C, and then N to C. Okay, so now that we have those two guys next to each other, we know that the minus here is going to interact with the plus there to make an amide or an amide linkage. All right, so how do we do that? Well, we join them. So how do we join them? Remember what happens? The only things that change are here and here. So we're going to have H3N plus, that stays the same. CH, CH, CH3. The R group never reacts. Then we link up the carbon with the nitrogen. That's the amide bond. There's the H. And then we're going to finish it out. The rest of it stays the same. CHH. Right? This is the R group. And this is the R prime group for gly, which is R prime, and val, which is R. And then you finish out the rest of the molecule. C double bond O, O minus. There is the amide bond. Carbon double bond O bonded directly to an N. Notice that this is N-terminal. And C-terminal. Now, why don't we call it O-terminal? Because the next linkage is going to happen at that oxygen, right? Just like we did it here. Oh, by the way, what left? This O left, right? And 2 of the 3 H's left. Plus water, so this is a dehydration. Okay. So there's the N-terminal. amino acid. There's the amino acid for valine, the R group. There's the R group, there's the R group for the glycine. And this is the C terminus on the right-hand side. And there's the amide bond. Good, that's val-gly. Now, how do we name these things? Well, it's pretty simple. You stick a YL ending for all of the amino acids other than the C terminus amino acid. We just use the full amino acid name of that. So we look at this and we say, this is the N terminus, so that's alanil. This is in between, I'll even call it middle. Any of the others is from glycine, so we call it glycyl. And since this is the C terminal amino acid, it gets the full name of serine. So alanil glycyl serine, shortened to alaglycer. That's not hard. All right, let's do a little more drawing here. Ser, thur, I don't know what you would call it, serine and threonine. Well, how do we go about that business? Let's see if we have this. In parts, or whether or not I have to do the whole thing, draw it all out. Either way, let's take a look. I forget whether or not it's loaded in or not. There it is. It's loaded in. So what's the very first step? We draw the 2. Remember, N terminal first, then carbon terminal, N terminal, then C terminal. You know that you're going to make the amide bond here. How does the amide bond go? This carbon bonds with that N. It leaves an H. And loses 2 H's and 1 O. There's the amide linkage. So that part stays exactly the same. And that part stays exactly the same. There's the water that gets formed, and there's the amide linkage. Man, you do this like just a couple of times and it becomes very automatic. Boy, I love automatic stuff. So cerylthreonine is the name for this dipeptide, or certhur. I don't know how they call it, thur. That works. A little bit like thank you, thur, may I have another. Anyway, let's keep going. All right, so for this guy, what do we have to do? We have to identify the 3 different peptides. Identify the 3 different amino acids. And once we identify what they are, we start from the left to the right and then determine what the 3-letter abbreviation is. So amide linkage, or amide linkage. So you have to identify the amino acid that's our group 3. That is our group 2. That is our group 1. And since we have this one here, that's sort of our group 0, right? It's a tetrapeptide. So that means there's 4 amino acids. So your job is to go find that list, be able to look through and figure out what those 4 amino acids are. What are they? Well, you look at the R group being just a methyl group, and that's alanine. You look at the R group that looks like that, and that's leucine. You look at this R group, that's cysteine. And you look at this guy, and that's methionine. And you'll know that met is the shorthand, cis is the shorthand, lu is the shorthand, and ala is the shorthand. So what we're going to call this? Ala, lu, cis, met. So that's the 3-letter abbreviation of this tetrapeptide. And if we wrote the whole thing out, we take alanine and call it alanil. We take leucine and call it Lucille. Now, B.B. King, who is was a famous, fantastic blues guitar player, and he had a famous guitar he called Lucille. This is not the same. Anyway, it's important. Cysteine, and then you write out the entire final amino acid. So that's the full name: alanil, lucil, cysteine, methionine, or ala, lu, cis, met, and you can see why they like the 3-letter substitution. Guys, that's not a bad problem. It looks like a crazy hard thing to do, but you're good at figuring out functional groups and then understanding what's in between, and then be able to match up patterns. I know you can do this. Practice up. Let's see, do I have the notes for this guy? Probably do. Hold on a sec. Oh, heck, that's the same one. So let's keep going. Oh, I see. We're going to go through the process. Good. So we chop, we chop, we chop. We identify the R group here as being alanine, the R group here as leucine, the R group here as cysteine, and the R group here as methionine. Not too shabby, right? Those are the names. We did it right. Good. All right, so let's talk in general about the structure of proteins. It's important. How they function in compounds is entirely related to their structure. The primary structure, the first thing you think of a protein making it different than other proteins, is the specific sequence of amino acids. You mix up the amino acids, you make a different protein, right? We already know that they're held together by peptide bonds from N terminus to C terminus, left to right. and it's considered the backbone of a peptide or a protein. All right? So there's the amide bonds. The peptide bonds are here, here. And so therefore this is glutamine, this is histamine, and this is whatever, proline, I think. Okay. Just some examples that are important. Insulin was the very first protein that they isolated and then pulled all apart and then determined what the exact primary structure was. Very important, because before they understood the idea of insulin, people would die from insulin deficient diseases all the time. And so the idea was to try to find either a substitute insulin or to determine and understand why people were sick with diabetes. Okay, so what's the story? They found that it has a primary structure of 2 polypeptide chains linked by those disulfide bonds that we had seen before in hair protein. It has an alpha chain, or an A chain. I guess it's not A alpha, it's A. A chain has 21 and a B chain with 30. And look, glycine, whatever that stuff is, valine, all the different ones. Amazing. That's a lot of work. Notice that we go from N terminal to C terminal in the end. Okay, so that's the primary structure of insulin. Which brings us to the whole idea of essential amino acids. They're all essential, but when we talk about the essential, these are the 9 amino acids that we must get from the diet, meaning they're essential as part of your diet. We can synthesize 11 of them. Okay? These are the guys. 1, 2, 3, 4, 5, 6, 7, 8, 9 that are essential because we must eat these. And they're found in meat and dairy. And then one, and that's the thing, all are in meat and dairy products. Why? Because meat and dairy come from other animals. And so we have similar amino acid requirements, I would say. But whenever we deal with grains and vegetables, there are different amino acid requirements because different proteins are being made. And so you can find one or more. And so what this absolutely means is that if you eat grains and vegetables and meat and dairy, you'll get what you need. But if you are vegetarian, and that includes vegan, okay, what that means is must be careful with your diet to make sure it, in varying amino acid intake. In other words, you got to make sure, look, if all you eat are beans and rice, beans and rice, beans and rice, beans and rice, you're going to miss out on some of the essential amino acids. So you have to vary your diet to make sure you're getting them all. Otherwise, it's not a healthy diet. Or you can also take supplements. But it's important that you're aware of that if you're a vegetarian or a vegan. There's an awful lot written about it. So if you decided to go to that lifestyle choice, that dietary choice, It's perfectly fine. It just makes sure that you're getting all the amino acids that you need to keep your body running at peak efficiency and that you don't end up with a deficiency. Okay. Now, let's deal with proteins and the secondary structure. Remember, primary is amino acid, amino acid, amino acid chain. That's the primary. How do all the amino acids hook up? Okay, in the chain. But the secondary structure is the three-dimensional arrangement of little parts of the protein. Once again, what are we doing here? We're starting to go from a very, very close look at the compounds with the actual chemistry, the atoms and ions involved to make the primary structure, right? Amide bond, which is carbons and oxygens and nitrogens. Okay, and then we sort of pull back a little bit and say, there's the amino acid, amino acid, amino acid. Now we're pulling back even more, and we're looking at localized region of a protein, and we're seeing how the interactions of the protein chains can interact to make a three-dimensional arrangement. They arise due to hydrogen bonding between the NH group, which is partially positive of one group, and the CO group, which is partially negative of a different part to the same chain. Okay, or a different chain. So the two stable arrangements that you get with secondary structure are the alpha helix and the beta pleated sheet. The alpha helix is most common, and it looks like a little bit like a slinky. It's a coiled shape held in place by hydrogen bonding. So you can see where the C double bond O with the lone pairs and the partial negative get close to the NH partial positive. And there's the hydrogen bond intermolecular force that sort of attracts one part of the primary chain with the other part of the other primary chain or within the same chain to make it sort of coil up. Shake hands in different spots, as you will. Okay, so the hydrogen bonds form between that partial positive H and the partial negative O of the amide bonds in the next turn of the helix. Now, beta pleated sheets are different. Beta pleated sheets is a secondary structure that has the polypeptide chains side-by-side arrangement. It has R groups above and below the sheet. And fibrous proteins like silk, they have these. So you can see that when they lay next to each other, we've got the N and the O, and there's the H, and there's the C. So partial negative, partial positive. Man, hydrogen bonding is huge, but you got two different chains and they're right up against each other, and they're not like straight. They sort of go in a, think of like an accordion. That's exactly how to think of it, like an accordion. Accordion, right? So it's folded over and then they're close to each other. Cool. So there's the alpha helix. You can see the coil part. And there's the beta pleated sheet. You can see how they lay next to each other. And then this is one protein, okay? It's one strand. see, how do we go? Coils in this area, right? A localized version area in the whole protein, and there's more, and then this swings around. Where's it go? Here, then here, then here, then more coils, then, and you can see where some parts lay flat because they're close to each other, and other parts coil up because they are able to get close with hydrogen bonding just in that spot. So these are all localized spots on the same protein. That's the key of a secondary structure. A triple helix consists of 3 alpha helixes chains woven together. Okay, think of a like a real strong rope. These always contain a lot of glycine, proline, alanine, hydroxyproline, and hydroxylysine, because there's lots of OH groups to make lots of hydrogen bonding so that they all can lay close to each other. Collagen is where you find a lot of these triple helixes. See the OH groups? So partial positive, partial negative. Lots of places. Okay. Collagen is the most abundant protein in mammals because of all of our structure and connective tissue. Tendons, ligaments, skin, plus the cornea, cartilage, and blood vessels. Man, that's an awful lot of our body. It's all of that stuff. Now, vitamin C helps stabilize those chains for whatever reason. And so whenever we are deficient in vitamin C, we have poorly formed collagen fibers. And as it turns out, when the fibers are elastic, our skin bounces back nicely, but as we age, the collagen becomes less elastic because there's extra cross links between the individual fibers called fibrils. And that causes problems like brittle bones and wrinkled skin, which is why all those extra creams and things that you buy at 3 o'clock in the morning on QVC and other infomercial sites, they're just sort of trying to avoid the inevitable, basically. This is just a natural process. So if we're looking at proteins and we want to say, well, what's the type of protein structure that is considered primary, or a beta-pleated sheet, or an alpha helix, or a triple helix? Would you consider a polypeptide chain held side by side by hydrogen bonds, and those are IMFs? I hope you recognize that if it's side by side, that's a beta-pleated sheet. The sequence of amino acids, well, that's the definition of primary. Corkscrew shape, gee whiz, it's not hard, but which one is it? Corkscrew shape sort of means helix, right? So which one is it? Well, I think 3 and triple is the big hint there. Not a problem. Moving on. So we have primary, which is amino acid sequence. We've got secondary, which is how they interact within the molecule to either curl up or lay next to each other. Tertiary. What's the tertiary structure? It is the specific 3D shape. We're pulling back even farther now, adopted by the entire peptide chain. Or in other words, once we assemble all the amino acids to make the peptide chain or the protein, okay, and then we put it together, and all the little parts that want to be close to each other and hydrogen bonds that are formed, they interact to fold up and make parts of the protein shake hands with other parts to the protein, and you get your secondary structure. Well, the whole thing then sort of wants to hold on to, get close to either, hey, there's an attraction there, or ooh, there's a repulsion there, to get the entire thing to look like a particular, well, it's sort of called morphology. It's basically how it all gathers up to make the shape of the entire protein. Hydrophobic interactions occur between two amino acids that have nonpolar R groups, right? So nonpolar are going to push away from the aqueous environment because this is polar, right? So in other words, it's a little bit like dissolves, only it's not really dissolves, it's more like gathers with. So like gathers with like. So this is a nonpolar R group, and this is a nonpolar R group, and they are going to like being close, but they're not going to want to be close to anything polar. That's hydrophobic interactions. Hydrophilic interactions, polar with polar. It's sort of like gathers with like again, right? So you're going to make partial negative, partial positive with the aqueous environment. So they're going to want to get pulled to the outer surface, right? Because it's in aqueous solution. That's what our body is, 90% water. There are salt bridges in which the minus and the plus attract each other, right? They're not going to bond, but they're going to get close because plus and minus are attracted. And then you have actual hydrogen bonding that forms because they get close enough to the H of the polar R group and the O or N of another polar R group. All of these things combine, and little spots along the protein molecules to, in the end, put the entire protein together into its tertiary structure. There's also disulfide bonds, which are actual covalent bonds that link parts of strands. Okay? It's a covalent bond. It's an actual bond. So the interaction of all these R groups on the amino acids put the thing all together. So these are all the amino acids that come together. And in particular, this amino acid has that hanging out. And then this amino acid right here has this hanging out. That salt bridge brings this part to this helix close to this part of this helix. Right? Okay. This amino acid has this part hanging out, R. This amino acid has that part hanging out, and that's even closer because, or another close interaction with a hydrogen bond IMF. Here we've got two beta pleated sheets that get close to each other. Here we've got something with a sulfur and something with a sulfur, and we make these disulfide bonds. So these two parts stay together in this place. Hydrogen bonds set up between the two beta pleated sheets to put those close together. This brings this part of the protein toward where the water is, right? Because these are polar. And then this part sort of stays on this side and together because these are both nonpolar. So they're going to maybe stay away from the water more. And this is what this particular protein always looks like, with everything in the same place every time, just because of all the interactions that take that long primary structure and make it look like that. It's pretty amazing. We do not. Well, this is what I'm going to do for you. What I'm going to do is I'm going to get you the right answers, okay? And then I want you to look at leucine and valine, try to decide what the R groups are and how they interact and how you might expect them to be. I'll give you a hint. Leucine, R group is nonpolar, and valine, R group is nonpolar. So knowing that, which one of those do you think it is? Cysteine, the R group contains SH. So which one of these do you think that's going to make? Aspartic acid, the R group has a O minus on it. It's one of the acidic R groups. And lysine, R group has a plus charge. So what do you think out of these four you might expect? Well, I hope it's obvious that when you have leucine and valine and you have nonpolar, that's hydrophobic. They don't want to be close to water. The two cysteines with two sulfurs, that's the only way that you can make a disulfide bond. And when you have a minus and a plus, that's an electrostatic interaction. Good. Again, that's just looking at those particular amino acids, looking at the R groups, and then deciding how the R groups interact, because that's what tertiary interactions, it's R group interaction. Determines what the tertiary structure of the protein will be. All right. Well, we had primary, secondary, and tertiary. Now we have quaternary. Quaternary structure is biologically active proteins with two or more chains or subunits that are stuck together. Okay, so we basically make a couple of tertiary proteins and then we combine them to make almost like a super sized protein. So hemoglobin, oxygen transport in our blood, has 4 polypeptide chains, 2 alpha chains with 141 amino acids and 2 beta chains with 146. All right, so there's a tertiary structure and a tertiary structure and a tertiary structure and a tertiary structure, and they like to interact in such a way that they make this quaternary structure. Right here is the heme group. All right, so there's hemoglobin. What's the story with hemoglobin? Each of the subunits is held together by those same interactions in tertiary structures, R group interactions, okay? And that holds each of the 4 different globes of hemoglobin together. And each subunit has an embedded heme group containing an iron center. The whole thing is heme, but that's the key. Okay, iron in your blood, that's what we're talking about. And that can bind an oxygen molecule. When it's not bound, it's blue, no O2 bound, but it turns red when O2 is bound to the heme group. So, summary of the protein structures, you have to keep it in mind, to keep it in your head. Primary structure is just the amino acid sequence. Secondary is these little spots along the chain where different parts gather together to make little special sections. Tertiary is whenever the whole thing folds into a 3-dimensional shape, where we take the beta-pleated sheets and stick them close to other beta-pleated sheets or into a place where it's hydrophobic, or we connect them with sulfur disulfide bridges, etc. And then the quaternary is 2 or more protein globs or subunits. Globs. You get the idea? Globin, hemoglobin. And that makes the biologically active protein. Very cool, right? Sickle cell anemia is sort of interesting. For one thing, if we look at a particular protein and we take a glutamic acid, which is what's supposed to be there, and it's polar, and we stick in the nonpolar valine, what happens is the interactions change. You start to form a different primary structure, which then can change the quaternary structure, and the red blood cells become elongated and crescent shaped. They look like a sickle instead of round. The shape interferes with the oxygen transport, makes people sick, and it's not good, can lead to ruptured capillaries, organ damage, and eventually death. However, 2 inherited genes cause this sickle cell anemia, okay? So it's a genetic disease. But if you have only one inherited gene that causes this problem, okay, what you have is a mild form that also, for whatever reason, provides protection from malaria, which is in Africa and South America a very deadly disease. So this is where natural selection is very interesting, okay? Because what is the whole point with inherited genes? What is the point of, um, you know, a species and an individual in the species? If you look at it just from biology, it's all about passing on your genes, making babies, and continuing the genetic line. So we've got a bunch of people getting together, and they're making these two inherited genes that cause this sickle cell anemia. People get sick and they die. Maybe they die early in life. Maybe they die later in life. Maybe they get sick early such that people don't want to make babies with them, and so they don't pass on the sickle cell disease very readily. Maybe they don't get it until later, so they don't know that they're passing along the gene, okay? If you only inherit one gene, it's a mild form. You may not even know you have it. You're a little bit sick, but if you get malaria, and lots of people get malaria from infected mosquito bites, then what happens? You die. You don't get to pass along your genetic code. So in a way, there's a natural selection that actually favors inheriting one of the genes, because then you don't get malaria. You live long enough to make babies. It's an interesting situation. And this is probably why sickle cell anemia still exists today, because one inherited gene individuals would continue to go on and produce babies that also had the one inherited gene. I find that pretty darn fascinating, just to see how things end up and why they might end up. Natural selection is very cool and interesting. And here it's absolutely related to biochemistry. There it is. So, do you want to have sickle cell anemia? No. Unless you're living in the Amazonian jungle and you would like to have a mild form of it so that you didn't get sick and die from malaria, which kills many, many, many every year without the medicines that allow you to recover from it. Cool. Okay, so now let's look at each of these and decide the level of the protein and structure that is related to beta pleated sheet. The beta pleated sheet, well, that's a secondary, right? It's within the same peptide. So it's secondary. The order of amino acids, well, that's primary, right? We know that. A protein with two or more peptide chains, two or more chains that are stuck together. Everything else except for quaternary is all in the same chain. And disulfide bonds is tertiary. Good. All right, now let's do just a couple of reactions. Protein hydrolysis, protein denaturization, or denaturation. Denaturing. I think I mispronounced that the last time I tried to say that word, denaturing. Okay, so protein hydrolysis, hydro, water, lysis, cut. So we are going to break or cut the peptide bond by using water to yield the individual amino acids. So in other words, we're just going to disassemble the protein, taking all the individual amino acids and pulling them all apart. This occurs in the stomach when trypsin or pepsin or other enzymes hydrolyze those amide bonds. That's what they do. It's the digestion process. Okay, so alanine glycylserine. So that's the ala part. That's the gly part. And that's the ser part. Those are the R groups that are associated with these. So hydrolysis comes and chops the amide bond. And remember, all of these are N, C, left to right, with the N terminus and the C terminus. So that allows you to remember how to do this. This part has to be here, and this part has to be here. So that's the alanine amino acid. There's the glycine part. And there's the serine part. So your job is not only going to be able to take the amino acids and put them together, but be able to look at a peptide chain and through hydrolysis determine what all the individual amino acids are. You can do that. It's just the opposite direction. Now, denaturing of a protein occurs when a change disrupts the interactions that stabilize the secondary, tertiary, or quaternary structure, not breaking the amide bonds. As you can see, we're not pulling apart individual amino acids from others. Basically, we're sort of straightening the chain. Straightening the chain a little bit. Understand how I'm thinking about that? The protein unfolds, and there's an overall loss of the tertiary structure, and the protein is no longer biologically active. Okay, one is, an example is cooked egg whites, right? What happens? They start off as clear, and then whenever you cook them, they do that solid, opaque white. Well, what you've done is you've denatured the protein. There's another way to do this, which is sort of cool. And what you can do is take some milk, okay, and put some food coloring in it. Okay, that's just so that you can see what's going on. Then you can put a couple of drops of dish detergent. That sort of slows things down. And then drip vinegar into it. Try this at home. Seriously, it's pretty cool. Because what happens is the vinegar, the acid goes in and denatures the protein. And it's sort of swirly because what's happening is that these proteins that are sort of all coiled up and curled up and crumpled up start to flatten out, and they move in there. And the food coloring gets pushed out of the way as it's moving, but it does it slowly through the soap. And so what you end up seeing is swirling. Man, if you're with kids, it is a really cool experiment to show because it's really neat looking. It's, you know what it sort of looks like? It sort of looks like the Aurora Borealis. Honest. Not that I've ever seen. It's one of my bucket list things, see the aurora. But it sort of swirls all around. Give it a go. It's an interesting experiment you can try at home. heated, all right, so that's the cook an egg. The heat disrupts those hydrogen bonds. Things are wiggling too much, because heat means kinetic energy, moving of molecules and bonds, and so the whole thing is sort of wiggling too far. The hydrogen bonds break, and when that happens, the hydrophobic interactions flip around, and it all sort of straightens out the chain. Now, it doesn't change the nutritional value of the proteins, but what would you rather eat, raw eggs or cooked eggs? So why doesn't it change the nutritional value? Because your enzymes cut the proteins into individual amino acids anyway, right? So if they're going to split them into individual amino acids anyway, all this does is sort of start the process and makes them, more tasty to eat, easier to eat, easily digestible. Hey, if you're already digesting by doing this first, it makes it easier later, less work to break the thing apart. So that's why it doesn't change the nutritional value, because it doesn't change the protein sequence of the amino acids. We also use high temperatures to disinfect surgical instruments, because it'll denature the proteins of any bacteria or viruses. Big deal, right? Big deal, because if you can denature the proteins of a virus, it's no longer going to be able to infect you. Knock on wood, hopefully. You can also change the pH. Once again, we're doing the same sort of thing, but you can also interact with those hydrogen bonds and those salt bridges. Tannic acid, a weak acid used to burn ointments, okay, is put on the site of a burn to coagulate the proteins, and it makes a scab, basically, which prevents loss of fluid from the burn and gives it a protective covering. And then there's organic compounds, ethanol and isopropyl alcohol, also disinfect by changing the hydrogen bonds from the protein to water to protein to ethanol. Okay, when that happens, it disrupts those side chains because it's not the same as water. And so any disruption means it changes the protein. And it's the same sort of thing. And finally, silver, lead, or mercury 2 can denature proteins as well by messing around and forming bonds with the ionic parts or reacting with the disulfide bond. And we do this with young babies. Give them silver nitrate into their eyes just in case any bacteria that could cause gonorrhea is present. It gets placed in there and it destroys that bacteria, so that won't happen. You can even do it by agitation, which I think is sort of interesting. The whipping of cream and the beating of egg whites into a froth is an example of using a mechanical agitation to denature the protein. Remember, those proteins are, the tertiary structure is just through not bonding, but through intermolecular forces, basically. And so the whipping action stretches the polypeptide chains so that those stabilizing R group interactions are disrupted, because once it's stretched, they're not close. And as a result, you make these, stiff peaks. All right, so how do we denature proteins? You heat it. You can add acids or base. You can add particular organic compounds. They're usually polar. You can add heavy metals, or you can actually agitate. Excellent. Okay. Hack at that. Do some homework. Practice hard. Good luck. Oh, before we're done, let's look at this. Sorry about that. You can still have good luck, but let's look at this problem. I wasn't thinking of how we were going to answer this. Let me find this in my notes. Hold on. There it is. Okay. So if we completely hydrolyze, do we really need to look at the notes? What do? Well, you end up making 3 different amino acids. Basically, you don't make them, but you sort of liberate them from the chain. So what are you going to make? You're going to make alanine, serine, and valine. Separate amino acids. That's it. And they'll be present in the Zwitterian form. With the N terminus over here plus, and the C terminus over here minus. Okay, now, tannic acid forms a scab on a burn. An egg is hard boiled by placing it in boiling water. What is similar about these two? Both are protein denaturing events. And what changes? Are we doing what we did up here? Separate into individual amino acids? Heck no. They break up interactions but only the interactions in the secondary and tertiary structure of the proteins. Okay? We are not hydrolyzing. Big difference. Okay? Excellent. There it is. Good luck. But before we go, I forget how many we go here. So amino acids and proteins. So a protein contains amino acids that have the N terminus and the C terminus, the ammonium group over here, and the carboxylate group that's over here, with the R group that's in between, up on top, right? That's what all the amino acids are. We stick the amino acids in a specific order to make a peptide bond. That's the primary structure. We add connections between them to make the secondary structure, which is the alpha helix and the beta sheets. Secondary structure makes it sort of look like that. And then tertiary and to give you the secondary structure, and then the tertiary, and then quaternary structure, sticking them all together. Okay. Proteins undergo hydrolysis to give you the individual amino acids, or they denature when you change the connections but not the amino acid sequence. So you keep primary structure. And whenever you do that, what do you do? Or how do you do it? You can do all this stuff. We can agitate it. We can react it with heavy metals. We can add organic compounds. Or you can heat it, or you can react it with acids and bases. All different ways to denature these proteins. I think we're done now. Good luck.